Starting?
Yes.
Okay, yes, we are starting. So hello and welcome to the next in our series of webinars at WSET School London. Hopefully most of you know by now that we've been doing these throughout lockdown, or since it began. And while we've not quite got our tastings up and running yet at the school, we hope to be continuing those soon. In the meantime, we have a very special presentation for you tonight, so I'm very glad that you could join us. The plan will be to go through the presentation. If you have any questions, please do pop them in the chat and we'll be addressing those later. That's as much as I'm going to say. I will pass it over to you guys to introduce yourselves.
Okay, I'll start. I'll just want to welcome everyone. It's a pleasure to be back on your screen. I've done this before, and it's even bigger pleasure because I'm working with together with Anne and Sam Harrop, who I know for over 20 years. He's a great friend and a great professional, and they agreed to. Well, I agreed to. We all agreed to be here to do this session for you. And I just to start with, I want to put everything in context and talk about the importance of microbiology from two perspectives. From my job as a consultant, in terms of production, in terms of communication, be it writing or speaking, and also judging wines, and also my job as a wine buyer. And there is a common thread between those two, and is a vitally important point to consider. We always have to think about the consumer, what the consumer really wants. And one level, all the consumer wants is something fit for purpose. And at the top end, the consumer wants something that displays just sheer quality. But at all price points, be it entry or middle or top, there is an importance of value. It's absolutely crucial, so the consumer comes and actually reach for the second bottle of wine. So as a consultant, and Sam will speak more about it, but as a consultant, the projects I do, I have to ensure that the process is reliable, the fermentation is safe, it's sound, there is no flaws, there is no wastage of wine. I want to respect the integrity of the grapes, to protect what nature gave us, and also to enhance their potential. We have to protect the sense of place, and sometimes this comes by avoiding any flavor deviation. So as a buyer, and that's a very important, I was trying to reflect in how I can explain the importance of microbiology from a buyer's perspective. And I'm going to use a comparison which most of us are familiar with. Most of us have heard of Maslow's pyramid of needs. It's just described of human motivations in several levels, be it physiological, in terms of having food and water and warmth, or safety, to be protected, or a sense of belonging. So this is us as humans. But as a buyer, I'm able to draw some parallels. First, I want to ensure that the wines are safe, they are sound, they are stable. I need to manage risks as much as possible. And all I want is to avoid any problems, to avoid a product recall. Because if you are involved on something like that, and I have in my professional career, it's very negative and it's very time consuming. So probably that's one of the most important things for a buyer. The most basic thing is to avoid any fault. But also need to avoid issues with shelf life, especially now that some products are moving very fast, some are moving quite slowly, and because of the pandemic. But the ones that are moving slowly, there is a problem. You want to make sure that the shelf life is protected. I think as a buyer, the most important thing is to provide value, and that could be either a wine that expresses the grape variety very well, or the site very well, or more important, you have to take into account that the wine is going to have to compare favorably against your competitors. I live in London and I work in London for many years, and it's possibly the most competitive market, one of the most competitive markets in the world. So I have to ensure that the salespeople that are working for the same company that I am, when they put the wines in front of a sommelier or a manager or a buyer, the wine has to stand out. And at the end of the day, all we want is to sell wine of great quality. But there are some of the most important points to take into account. And I said to you guys before, I think the microbiology can help, but one of the most important things you can think is to have the consumer always on the forefront of your mind. The consumer is different. The consumer could be just a consumer of entry-level wine, and their expectation is for something fruit-forward, harmonious, pleasant to drink, without any fault. They don't want to know about grape varieties, about blends, about vinification techniques. All they want is to have a nice wine in their glass. And microbiology is important in this case because we're talking in most cases with big volume, and from the production side or from the buyer side or from the consumer side, we all, in this case, microbiology can help because to avoid risks. In the mid-premium sector, we want wines that have a sense of place or have variety typicity. And microbiology is very important, and Anne is going to speak about it, on how yeast and bacteria can help to accentuate quality as well as the sense of place. And at the top end, right at the top of the tree, what the consumer wants is a wine with exceptional quality and a story to tell. And I know some of the best wines in the world, they do use microbiology just to be safe. In general terms, we don't want to be adding anything to must or to wine unnecessarily, but also is unwise to leave things to nature in most cases. So to have the knowledge and the ability to apply science, I think is pretty vital. And I'm sure Sam is going to speak about sympathetic winemaking when he drills down to more technical details of winemaking. So I'm really excited to hear now what Anne is going to tell us. But from my perspective, as a consultant and as a wine buyer especially, microbiology has a very, very big part to play. And so over to Anne.
Thank you, Junior. Sorry about that. I'm sharing my screen and I can get started. Okay, so if there's a problem with the screen, just let me know or let Julia know. She'll be able to send me a message. Thank you, Junior, for this, for putting the topic in perspective. My name is Anne Dumont. I'm a microbiologist. I work at l'AROMA, who's a yeast and bacteria specialist. Like I said, I'm a microbiologist, but I also did my enology degree in Oregon, who is sadly now burning like crazy. Some of my favorite places are going up in flame. It's really sad. And where I studied the impact of wine yeast on Riesling, Pinot Noir, and Chardonnay, and that's where I fell in love with wine. And I think it's a very good place to fall in love with wine. And I'll be talking today about my favorite bug, which are wine yeasts and bacteria. I'll do a very quick winemaking overview. I don't think I need to teach you anything about winemaking, but really just where wine yeast and bacteria are important in the process. And I will talk about their role in winemaking and how they contribute to wine style. And I'll also want to say hello to one of my colleagues who I just saw is part of the participant. Her name is Sibylle Krieger. And Sibylle is probably right now the top wine bacteria specialist in the world. Her experience, her studies, and she selected a lot of wine bacteria. So if any of the questions on wine bacteria are very precise, I might ping you, Sibylle, to say something about that, and we're really lucky to have her with us. So anyway, let's get started. And what I really like to do during those presentations, and that's one of the problem with doing them virtually, is that I can't see people, and I like people to participate in the discussion and ask questions and challenge us, and you know, this is a place for learning, and unfortunately this doesn't allow us to do that. So what I'll do at the end of different section, I'll stop and answer a few questions that Julia will send my way, and then answer more questions at the end. And if there's some questions that we didn't have time to answer, you have my email address in the presentation. Don't hesitate to email me, Sam Junior, and we'll be more than happy to answer all your questions. And like I said, there's no stupid questions, there's no bad question, there's only questions to learn and to progress into the information related to wine yeast and bacteria. So in the winemaking process, and here I've just put red and white grapes because it could get complicated, rosé and sparkling, but we can use wine yeast, of course, for the fermentation in the juice and the cap for red wine. And of course, we ferment at higher temperature compared to the fermentation of the grape must in white wine, and also at lower temperature. And Sam maybe can explain a little bit why during his presentation. And of course, it is followed by malolactic fermentation in reds, and in wine, sometimes you do malo, other times you don't do malo. And in the last 10 years, and even more than that, we are now doing co-inoculation, which, and I'll talk a little bit more at the end of the presentation, that we do alcoholic fermentation and malolactic fermentation together in both red and white wine. And it is a stylistic decision, and it is also a bioprotection situation to protect the wine from the development of spoilage microorganism. But anyway, I'll talk more about that when we get to the wine bacteria section. So let's get started. There's a very simple equation in wine, in that in order to make wine, you need two things. You need grapes and you need yeast. So if you don't have that, you don't have wine. You have grape juice that is turning into vinegar or into something that doesn't taste like wine. And the wine yeast that is responsible for the alcoholic fermentation is Saccharomyces cerevisiae. So it will use the grape sugar, glucose and fructose, which are the main sugar, and transform them into ethanol and CO2. The other microorganism is responsible for malolactic fermentation, is called Enococcus uni, but there's another one that we're now using, and that I'll talk about later in the presentation, that is called Lactobacillus plantarum, and it will convert the harsh acid, malic acid, into lactic acid and CO2. And all this is influenced by many factors, and that's going to determine the style of the wine, which is, of course, grape variety, the grape precursors. And the grape precursors are the non-volatile aromatic compound. I call them potential aromatic compounds that the yeast will transform into volatile aromatic compound that you'll be able to smell. And that is through the action of wine yeast. And of course, there's the vineyard and the winery condition, the microflora, the hygienic condition, the phytosanitary treatments of the vineyard that will all influence what's happening with the microorganism. Of course, the production of the yeast metabolite, it doesn't only produces aromatic compounds, but also other compounds. I'll talk a little bit more later. The winemaking techniques, if you're doing maceration, fermentation, temperature. So all those are important in the determination of the style of wine that you'll be making. And of course, the aging of the wine. So those are all the factors that will determine the final product. So yeast and bacteria, let's dive into the topic. Yeast and bacteria are completely different from one another. The bacteria that, and I think I can do a pointy thing here. I don't know if you see that. The bacteria that we are interested in are gram-positive bacteria. And they are prokaryotes. The wine yeasts are part of the eukaryotes family, and they are in the fungi family. And in more details, the prokaryotes, the bacteria, and the yeast, which are eukaryotes, are quite different. The bacteria don't have a nucleus inside their cells, whereas the yeast do. The bacteria are a lot smaller than yeast. And of course, the species that we're interested in are Eunococcus, Lactobacillus plantarum, and for the yeast, the Saccharomyces cerevisiae, and many others that, and I'll be mentioning some Latin names to confuse you a little bit during my talk. And the actions of the wine bacteria in wine is via growth and transformation, and the action of the wine yeast are growth, respiration, fermentation, and transpiration. So they're quite busy when they are in a grape must. So where do microorganisms for wine fermentation come from? And this is where we'll be talking about some philosophical divide between spontaneous and inoculated fermentation. That's going to come a little bit later. But let's understand a little bit how the microbial population in wine is very dynamic. So during the winemaking process, there's a succession of microorganisms that are present in this environment. Some of it is good and others are not so good. So for example, on the grapeberry, we have a lot of microorganisms, and the size of the bubble indicates how, in terms of number, how present they are. And you can see on the grapeberry, the most predominant species are non-fermenting, mainly oxidative flora. And you don't want that in your must to ferment because it's going to produce faults in your wine. And you have some fermenting species, but they're not very tolerant to ethanol, and they're quite sensitive to SO2. So they're not going to be able to carry out alcoholic fermentation. And once you press the grapes and you destem them and you have must, then there's a switch into the number of the predominant species that are present. So you see the non-fermenting species, the oxidative flora, is quite reduced. There is more fermenting species, the slightly one, and there's an increase in the fermenting species. This is where you'll find Saccharomyces cerevisiae that are more tolerant to ethanol. They're less sensitive to SO2, but they're still sensitive to the nutrient content. And this is when we need to measure the YAN, the assimilable nitrogen in wine in order for the yeast to thrive and perform alcoholic fermentation. And during fermentation, this is where you see that the wine yeast are taking over. Saccharomyces cerevisiae is really taking over the fermentation, and it's going to carry through, if everything goes well, until you get the complete fermentation. And when the fermentation is finished and the wine is aging or maturing, then you see you don't see any of the non-fermenting species or the species that are intolerant to ethanol and SO2, but you see more resistant species, and this is where the baddies occur, and I'm talking about Brettanomyces, which are very tolerant to SO2, to ethanol, and also to oxygen deficiencies. And so this is where bad things can happen, and hopefully they don't. But this is the an illustration to show you that this is not a static environment, but it really evolves depending on the process.
And if I can just jump in there very quickly,
Yes.
Anne, I think
Yes.
the slide provides a fantastic illustration of obviously of the different yeasts involved in the process. But the point I would just reinforce, if we go back to that slide, Anne,
Mm-hmm.
Could you go back to that slide?
Yeah, I'm there now.
Yeah. The point I want to reinforce here is the size of that green circle under grape berry, okay? Now, you've said it already, but just to re-emphasize that the Saccharomyces cerevisiae is not, there's not a lot of it on the grape berry. It's a small population. It's actually really, really hard to find on the vine. And so when people talk about spontaneous fermentations, indigenous fermentations, primarily they're talking about this, this section here, the non-Saccharomyces at the must stage. And the reality is that the vast majority of those ferments don't actually, the vast majority of ferments don't actually allow for those non-sac yeast to get a chance to get a foothold in the fermentation. And so what's happening from start to finish in the vast majority of indigenous spontaneous fermentations, I should say, is that the Saccharomyces cerevisiae that predominantly lives in the winery is doing the work. So it's not actually, the vast majority of spontaneous ferments are not indigenous ferments. And that's a really important point to raise. Be careful what you believe from passionate growers who are passionate about indigenous and spontaneous fermentations. It's a great concept. The vast majority of spontaneous fermentations are not indigenous.
Thank you, Sam. And like I was, that is very interesting and indeed very true. And I'll get into a bit more detail about that. And what Sam is saying is really interesting. And I think I have a slide that's going to illustrate that a little bit later on. And I'll go into more details about that. And just to show you, and I love to throw Latin names of yeast around because it makes me sound really smart. Of course, Saccharomyces cerevisiae. Saccharomyces, it means sugar. Myces is for the fungi, and cerevisiae is actually a Latin name for beer. So, and you'll see why later. And other non-Saccharomyces that we will find in either on either the grape berries in the vineyard or in the must are called Candida stellata, Cluveromyces thermotolerans, Enseniaspora ovarum, Pichia membra nifesciens, Brettanomyces bruxellensis. That's the baddie that everyone knows. But there's also other non-Saccharomyces that are very good that I'll talk about later on, the Meshnikovia family and also the Lachantia. So I'll get to that into more details in a little while. And yeast Saccharomyces cerevisiae is probably my favorite microorganism simply because it makes the best foods, the best fermented foods in the world. And of course, there's beer. That's where the cerevisiae comes. Bread, sake, of course, wine, but also the fermentation of chocolate and coffee. Some they can be used for probiotics. Some species of Saccharomyces are used as probiotics. So basically, the good things in lives come from Saccharomyces cerevisiae. And I'll be talking about the notion of strains because they're all the same species, Saccharomyces cerevisiae, but when we talk about different strains, it's like we're all human beings, we're all the same species, but each one of us is different. Each one of us is a different strain, and so we're all unique. We all have different capacity, different physiological characteristic, and that is based on where we come from. This is our genetics, but also it comes from our environment and how we are shaped by what we eat and what kind of activities that we do and so on. And so it's the same thing for wine yeast. So it's all the same species, the main fermenting species, Saccharomyces cerevisiae, but they are so many different strains of them. L'Alma is one of the largest private wine yeast collection in the world, and we have over 10,000 different wine yeasts in our collection. And in production, so that means commercially available, we have over 200 different wine yeasts, just to tell you. And they're all different from one another, and they all do different things, and they can be used for different grape varietals, for different technological characteristics. Some of them are better used in red or whites or rosé, and some of them are used for sparkling wines. And there's also the flora yeast, and we don't produce any of that, but there's the flora yeast that are used for example in the Jerez production. So the biodiversity of Saccharomyces cerevisiae is so rich, and we're still learning so much more about them every day. The more we study and isolate them and understand what they can do, it's a very rich environment. And what happens during the succession of yeast during indigenous fermentation, and that also happens when you inoculate a wine must with selected Saccharomyces cerevisiae, is that at the beginning of fermentation, there's a peak of non-Saccharomyces increasing, and as the alcohol level increases, Saccharomyces cerevisiae will take over, multiply, and carry through alcoholic fermentation. And this is in the best case scenario. This is what you want your fermentation to be doing. You want Saccharomyces cerevisiae to kind of bully through the fermentation and take over in order to let, well, to finish the fermentation and to bring it to dryness, if that is your goal. And you also want to prevent the development of bad non-Saccharomyces cerevisiae, because some of them will produce a faulty aroma compound, and that is a big problem. And just to illustrate what it means, at the beginning of fermentation, then you have a lot less Saccharomyces cerevisiae, and you have also non-Saccharomyces cerevisiae. And at the end of fermentation, then you have more Saccharomyces cerevisiae and non-Saccharomyces cerevisiae in less number and almost non-existent. And what you want to do actually when you inoculate your wine must with a selected Saccharomyces cerevisiae is that you don't want the non-Saccharomyces cerevisiae to eat all the nutrients and don't leave any for the Saccharomyces cerevisiae, and you want a rapid population development of this microorganism in order for the fermentation to start really well and to finish really well. That way you decrease the risk of volatile acidity and also sensory deviation. And I'm going to talk a little bit about the indigenous fermentation and the risk of spoilage, and and I will let Sam debate about this if there are some questions on this topic, because there's a big philosophical divide about whether this should be an indigenous fermentation and whether or not it represents more the terroir. And my take on this is that, first of all, the notion that Saccharomyces cerevisiae is found in the vineyard is very sketchy, because what does Saccharomyces like? It likes sugar, because that's what it's going to eat in order to reproduce and multiply, but also to do fermentation. And in the on the grape berry, if the berry is not broken and it's healthy, then you don't find sugar on that. So for them, it's not advantageous for them to be in that environment. It wants to be in an environment where it's going to eat as much sugar as possible, because this is how it's going to survive. And so that's why, and Sam correctly mentioned that, is that that's where you'll find them, in an environment where there's a lot of sugar, and that is in the winery. And they will associate on winery equipment, on bees and all sorts of insects. Also, they've been found on birds. It is advantageous for them to be associated with human activities, because we can bring them to that sugar. So it's really a human friendly, maybe the first domesticated living thing, working with humans to produce all those fermented food, but that's what it does. So to me, when they say that it represents the terroir, then I'm always questioning this. And you know, it's a question of preference, whether you like wines that are fermented spontaneously or that are fermented with selected wine yeast. And for me, personally, I have some problem with some of the faults that we find in spontaneous fermentation, because those funky wine aromas don't represent the grape varietal, the origin, the recognition of the grape varietal. But that is a personal choice, and other people might like that, and that is fine. But there is a risk of spoilage, and one of my colleagues was referring to spontaneous fermentation as a roll of the dice. And some winemakers, you know, I think when they do spontaneous fermentation, they really are carefully monitoring, sleeping beside the tank kind of thing, to make sure that there's no serious deviation. And kudos to them for taking that extra care. And so it's really a question of preference, but I don't think that when you use selected yeast, you don't have a wine that is not specific to the terroir. And I would argue the contrary, that it does indeed respect the grape varietal, because it will really optimize its potential. So I think, Sam, you might want to comment on this.
Yeah,
I can comment. I just want to make one point, and
yeah,
because it concerns just something that happens to me, and I was quite surprised. I was in Bordeaux. I'm not going to name the winery, but I'll make you guys guess. It is one of the top wines in the world, and is made in the left bank of Bordeaux. And I was walking through the vineyard and the winery, and I had a very open conversation with the winemaker. And we're talking about fermentation, and he very openly said, "No, I use I inoculate." And I was surprised. So I thought wines that they wouldn't. Many do, but they don't like talking about it. But he was very open about it. He said, "No, I use it." And I asked him what he uses and why. He said, "Well, he told me the yeast he uses, and it is one of those microorganisms that doesn't detract from the fermentation, doesn't add anything. It just respects the terroir and converts sugar into alcohol." And so I was quite, even at the top end. And I asked why not say, "Listen, I'm talking. Our wines are extremely expensive, and it's too risky for me. And I don't, as you said, I don't want to roll the dice." Mm-hmm. So I'm going is one of the 5 best wines from Bordeaux. You can guess which one, and maybe Sam also has another point to make. But I thought it was interesting.
Yeah, we could go on about that for some time, but I'm just going to going back to that previous slide, and I'm going to raise a question Tim Bond has made, which I think is a good one. Do non-sac yeast bring any positives at the beginning of fermentation? And well, my answer to that, my view on that is they can indeed bring a lot of positives at the beginning of fermentation. We see some of these non-sac yeasts can provide more texture, more weight. Some of them can produce high levels of esters, aromatic compounds. You've got that word there, funky, that means different things to different people, but it's all about balance, right? And where non-sac yeast can actually work well, it's adding sort of another layer of complexity, another layer of intensity, but it's all about balance. And so, you know, you can actually inoculate with non-sac yeast, and this is growing sort of area of technology here for winemakers. And I certainly inoculate a number of my wines with non-sac yeast because they bring positives. One then overseeds the non-sac with Saccharomyces cerevisiae strain that will do the job that you want, whether it be stylistic or, as in Junior's case, in Bordeaux, more pragmatic, so just getting to dryness, sort of whatever the agenda is for the winemaker, and there are many. But yeah, to answer your question, Tim, non-sac yeast can be beneficial. The reality is, if they're not controlled, like anything that's not controlled, it's a bit of a roll of the dice, and it's a lucky dip, so you don't know what you're getting in it. In that instance, non-sac can bring a negative consequences to the wine making as well. But just one final point, and I think it's an important one I'll return to, perhaps if we have time, and mention, you know, this indigenous approach, or the spontaneous approach, being an ideological one, and I completely agree with that. I think there's nothing wrong with a spontaneous strategy, as long as the winemaker employing it is recognizing that it's not necessarily going to give you a great deal of control, and the results are therefore going to be spontaneous and a little random, and it's an ideological decision, but you're giving up a fair whack of the winemakers want control over the process. So you know, it's a lucky dip in a sense of as to what you get. Okay.
Very well said, Sam. And so, and I'll talk more about non-Saccharomyces, selecting non-Saccharomyces to bring their contribution at the beginning of alcoholic fermentation, because it's relatively new and very interesting, and I'm coming to that. Julia, was there any other questions that I could address at this point before I start going into the selection process?
Yeah, we've had a few different questions. I think some of them have been answered already. I might just pick out a couple quickly before we move on. And we might have to save some for the end, because I don't think we're going to have time.
Well, maybe I can answer one question, and then we can move on, because I'm already running a bit late.
Okay. All right, let's just go. There was one from Alessandra about some wineries say they wash their grapes before fermentation, presumably to try and control the different yeast populations. Do you think that is possible? Will it have an impact?
Sorry, say that again. That's if people would wash their grapes before pressing them.
Yeah.
Well, then you bring water into your wine. I don't know, Sam, from a winemaking point of view, what do you think?
I think the question was more directed at, is it going to reduce, is water going to be a sufficient process to eliminate the indigenous flora to give you more control during fermentation if you decide you absolutely want an inoculated strategy? Yeah. And I think the answer is yes, it will most certainly reduce the non-sac yeasts and whatever Saccharomyces yeasts are present.
Are there. Yeah, it's like if it rains before harvest, then you're changing your population dynamic into certainly for that. But when you're inoculating with Saccharomyces cerevisiae, they'll they'll quickly take over, and so they'll be able to control that indigenous flora without you having to, you know, wash your grapes with water.
Yeah, and
I think this question was probably related to I heard of this. I think it's probably related to a sparkling wine producer in Italy, if I'm not mistaken.
But if I could just jump in, raises a very, very good question, actually, and maybe we're digging too deep, but so it's very important that you understand that every growing season, obviously, with every growing season being very different climatically, the microflora that you see on the grapes is going to be different from one vintage, one growing season to the next. So any producer who says, I have an indigenous strain of non-sac or Saccharomyces cerevisiae that loves my vineyard, and I, you know, it comes from the vineyard and it gives me this character year in, year out, you know, the reality is that's just not true. That is just not true. Because every growing season is a different mix, a different balance of microflora. And as Anne's already pointed out, the winemaking yeast, the cerevisiae, is likely to be very, very low level. So these other non-sac yeast, then one year there may be a strain or a species that is very prominent because the weather has been very humid. The next year it may be completely different because of, you know, a dry growing season. So it's not as simple as saying my vineyard yeasts are the same from one year to the next. It's just not the case.
And also, you also have to keep in mind, there is no way the wine yeast that are indigenous to the vineyard and the winery, those wine yeast, they really, really don't know what kind of wine you're making, whether it's white or red, or you wanted a wine for aging or for early release, or they don't know what kind of wine you want to make. So stylistically speaking, it's a bit more complicated. And this is a bit of a summary about the advantages of selected wine yeast. They control, of course, spoilage microorganism. They respect the varietal and the terroir, and they can help develop particular wine style, or if, for example, there's very high sugar or high acidity, they can, you know, help overcome some of the problem associated with that. And now I want to dip a little bit into the process of selecting wine yeast, because it's quite interesting. Well, for a microbiologist like me, it's quite a long and rigorous procedure. It can take between 3 to 5 years, but sometimes it is as long as 10 years. We have 10 years. We have a wine yeast that has been part of a selection program for the last 10 years. And so it's quite a rigorous procedure, and it's often associated with a particular terroir region or for a particular wine style. And it is also in collaboration with local wine research institute and winemakers, because they are the teams that know those regions and the wine styles associated with those regions the best, or a grape variety. And when we start a selection, program based on a particular need. For example, there's, and I like to give that example because it's one of my favorite regions. We have a yeast that was selected in the Priorat region in Spain. And so the winemakers in the region were asking for wine yeast that were that would respect, of course, the different grapes that are present, and also the style of wines that is made there. And when we start a selection program, we don't go into the wineries just 1 year. We go over several years to have a large a large number of microorganisms, because like Sam was saying, it varies from one year to another, and we select them based on different criteria. So there's the technological criteria, such as alcohol resistance, temperature, lag phase, whether it is can implant very easily or not, and also whether it's compatible or not with bacteria for malolactic fermentation. And of course, the classical parameter of VA production, SO2 production, etc., but also style expression. And that's probably the most important part, is whether or not it can convert different potential aroma compound into the volatile aroma compounds, such as esters and thiols and terpenes, whether or not they can influence mouthfeel, because some of them will produce a lot of polysaccharide that will influence the mouthfeel and glycerol production, but also avoiding defects such as sulfur compound production and acetaldehyde production. So those criteria are part of the cahier des charges when we select a wine yeast. And what happens is that we go we sample grapes, we bring them back into the wineries, we put them in vats, and we let the fermentation run its course. And then we select those microorganisms on plates, and we do the yeast colonies analysis. And now that we have DNA fingerprinting, we can do their DNA fingerprint and make sure that they are unique, that they have the right species, separate those that are similar from one another, remove the bad yeast from the good yeast. And that can take 3 years to do that. And after that, we maybe end up, when we do that, maybe we have 1,000 different colonies. And after the 3rd year, maybe we have 300 colonies. And from those 300 colonies, we'll do a few quick tests and test their characterization, and maybe end up with 10 yeast that are potentially interesting. And then we'll do micro-vinification and taste the wines to make sure that they represent the style of wine that respect that particular terroir. And then we can grow the yeast in big fermenters that resemble a tank when you ferment wine. And then we dry them and we make them available to winemakers. But before we do that, we give samples to different winemakers in different wine regions to be able to characterize them on different grape varietals, because sometimes some yeast that are selected in a particular region will work particularly well in other grape varietal also. So that can be interesting also for winemakers to trial. And yes?
Time is running away from us, but there's one very important point I want to make, which is, I saw a question of someone just passing by. Everything here, we're talking about stuff that comes from nature. There's no GM, there's no genetic modification, and that's one of the questions. So everything is from nature, and in most cases, I don't know if all the varieties, in the vast majority of the cases, we use those yeasts for organic wine production. So everything is as natural as it can be.
Exactly. And like you said, we don't have any GMO yeast, and the terroir for the biodiversity is so large that right now we're plenty busy with what we have. And so this is a quick summary of during the fermentation, the different action of Saccharomyces cerevisiae on the production of higher alcohol and esters and organic acid and glycerol, and of course ethanol and CO2. And this is, I like to show that diagram because it really shows how wine yeast influence the sensory profile of wine. And it's really interesting. And like Junior said, time is running on us, so I'm going to accelerate a little bit. But each wine yeast strain, when it's selected, it is different from one another. And those wine yeasts, within their metabolism, they have different enzymes within their system that will produce or transform some compounds in different concentration. And the different concentration of those compounds in the wine, their interaction with one another, with the alcohol, with the different acids in them, create a unique profile of the wine. So when people say, oh yes, if I use wine yeast and my neighbor used the same wine yeast, but he has a different grape potential, the wines are not going to be the same. They are going to be completely different because the precursors are different, but also the activity of the wine yeast based on those precursors will also be different. So it really creates a unique product. And I'm going to talk a little bit about the contemporary technologies that are being used today, and Sam alluded to it earlier about the use of non-Saccharomyces cerevisiae. And you can leave it to chance, but now more and more we're selecting different species of wine yeast that cannot, and they're all selected during wine fermentation, but at the beginning of the fermentation. And so some species now are found for winemakers to use at the beginning, and each one of them will influence the wine style. For example, this Lachaussea thermotolerance, and here I have a few examples, will increase acidity in wine. So it's really interesting with global warming, where in hot climate condition you have less acidity, so less freshness. And when you use that yeast at the beginning of fermentation, then it increases the acidity. And then after that, you inoculate it with Saccharomyces cerevisiae to complete fermentation. Thiol production, everybody knows about Sauvignon Blanc and the typical grapefruit, passion fruit aromas, and yeast is completely responsible for the production of aromatic thiol. So without, and if you use different wine yeast, then the level of those different thiols, whether it's the 3MH, 3MHA, or 4MMP, will vary depending on the wine yeast that you're using and depending on the style of wine and the potential of your grapes that vintage. Then you can pick the right yeast in order to optimize the style of wine that you want. Some of them, for example, Torulaspora del Bruckheim, here in Canada, ice wine country, a lot of winemakers will use it at the beginning of the fermentation of ice wine because it will reduce VA production in ice wine, and it will contribute to the mouthfeel of the ice wine along with the residual sugar. But it's really, really interesting microorganism to use in conjunction with Saccharomyces cerevisiae, and also other species like Meshnikovia fructicola and Pulcherrima can help in terms of biocontrol. When winemakers have a strategy for low SO2 solutions for their wines in order to cater to those types of wine, then they will use those microorganisms because they have the possibility, they're using those microorganisms to reduce the impact of the spoilage microorganism because they will populate the environment. And after that, we have selected yeast that are specifically selected to produce very low SO2, H2S, and acetaldehyde, which is an SO2 binding compound, in order to produce those low SO2 wines. So they're very good and natural solution to produce stable low SO2 wine. So it's quite interesting to have those microorganisms to face the winemaking challenge. And let's not forget that with global warming, then those issues are becoming really weird, really real and not weird. And so just to show you a quick example, how different wine yeasts differ in their possibility to produce SO2 at the end of fermentation. So it's quite significant. And of course, I alluded to that earlier on, is that wine yeasts are really important in defining the wine style of the wine. So a lot of winemakers will use that in order to have different wine style and either blend them afterwards or sell them as such. But it's quite interesting depending on their the potential of their grape at that particular harvest. Then we can recommend wine yeast cater for their wine. And so moving on to malolactic fermentation, maybe I can take a question, or maybe Junior or Sam, you have some comment before I go on?
And I think, honestly, I think we better crack on, because we'll probably leave Julie will agree with me.
Okay. Okay, and we can take some questions afterward if there's some time. Malolactic fermentation, Sibyl, I will try to do you proud. Malolactic fermentation is the conversion of the harsh acid, malic acid, into lactic acid. So it's really important in red wine and also in some white wine, because you will reduce the wine acidity, so increase the pH and reduce the acidity. It also provides microbial stability to the wine, because it will eat the remaining carbon sword for potential spoilage yeast and bacteria. Spoilage yeast is Brettanomyces. And also we know now more and more that the metabolism of malolactic bacteria can really influence the aroma compounds produced in wines. And the lactic acid bacteria, which is the group of bacteria in which wine bacteria are found, are one of the most important food group and beverage bacteria. You think about cheese and saucisson and all the and olives, for example, they are all due to lactic acid bacteria. And the main bacteria found in must and wine are Lactobacillus, Leuconostoc, Enococcus, and Pediococcus. And the ones that are of interest to us are the Lactobacillus and Enococcus. And there are two species. LAB means lactic acid bacteria used in winemaking, Enococcus oni, which are very tolerant to low pH, low temperature, and also some of them are resistant to high alcohol. And now I'm having a little doubt here, and the Lactobacillus plantarum, which are used more in higher pH wine above 3.5, and also they can be used at higher temperature. And the wine bacteria are quite interesting, because it's more than the transformation of malic acid into lactic acid, but also it can shorten the duration of the winemaking process. If you inoculate with selected wine bacteria, then you don't have to wait for it to occur spontaneously, and sometimes it can be a real problem. So that can help to rationalize costs, because you don't need to heat up your tank to start the spontaneous malolactic fermentation. I also told you that it controls spoilage population, especially in Brettanomyces. There was very good research done by Vincent Gerbeau with his team in Burgundy that showed that inoculating with wine bacteria really controls the development of Brettanomyces in wine, and also the production of ethylphenol, that very unpleasant leathery band-aid aroma associated with Brettanomyces. You can lower the acetaldehyde content, so it's better for SO2 management. Also, it prevents the development of biogenic amines, and biogenic amines are allergens that are produced by spontaneous wine bacteria, and some of that allergen you'll recognize is histamines that can create an allergic reaction, but also other compounds like cadaverine and putrescine, and I'm pretty sure that the names are quite evocative of the kind of aromas that they generate. So by inoculating with wine bacteria, you prevent the development of those allergens. And also you can reduce your carbon footprint when you do co-inoculation of wine and bacteria, because you, and it's related to rationalizing costs, because you don't need to heat your tank, so energy saving. But also it's the sensory management. Everyone has heard of diacetyl, which is that buttery aroma, and that buttery aroma is directly linked to the metabolism of wine bacteria. It can also help with aldehyde consumption, aroma biosynthesis like the esters, very important in red wine, stability of color, and also mouthfeel development, and of course avoid aromatic faults like we were talking about cadaverine and putrescine. Then that's an important thing to avoid. And again, like the wine yeast, just to show the complexity of the action of the different wine bacteria in the production of aroma compounds, and not only the conversion of malic acid into the lactic acid. I really like that picture. I think it's really nice. And like we were saying, it really influenced the aromatic style of the wine. I think that for some people that are just starting to know wine bacteria, this is a very interesting topic, because when we do tastings of the same wine fermented with different wine bacteria, you really see the different sensory contribution. There was a very interesting work done at the AWRI in red wines, and to show that the different red fruit esters, aromas like raspberries and blackberries that are influenced by the different types of wine bacteria is really interesting. And a lot of decisions now I made for stylistic reason, depending on the wine bacteria used. And finally, now more and more people are using wine bacteria in co-inoculation, and the co-inoculation is the inoculation of wine bacteria at the same time, or 24 hours, I should say, after yeast inoculation, in order to, and it's a stylistic preference, so you're going to have less diacetyl produced compared to sequential inoculation, when wine bacteria are inoculated at the end of alcoholic fermentation. And the sensory profile will be completely different compared to co-inoculation. So those are the results of new research that are really helping winemaker, for example, in early releasing wines using co-inoculation can really be a good tool in order to complete both fermentation as quickly as possible. So I apologize. I know I ran through the last part, and hopefully that gave me a bit of time to answer some questions, or some questions for Sam and Junior.